This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 24, recorded December 30th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Happy New Year, Vincent, and to all our listeners.
You too. How's it been going this week?
It's been relaxing. The end of the semester and the beginning of the next has transpired, so we're ready to start the spring term on Tuesday.
Wow. Now you do medical school, right?
Yeah. And so we start early, and we have Maymester and all those other mesters. And
do you do a whole course yourself, or a big part of it?
Yeah, I do the majority of dental microbiology. The only pieces I don't do are the virology. I actually do the parasite portion, and I do all the bacteriology, and I have one colleague do the mycology, and then I beg to have folks do the virology for me. Mm-hmm. That's only because I would collapse, I think.
I have just 4 lectures that I give in our medical microbiology course here, and that's not for a few weeks. And my virology course begins the 3rd week in January. That's an undergraduate course, so I have a few weeks left.
Oh.
But I'm getting ready for that. That's a good one. It's a lot of fun. I enjoy doing that.
Yeah, the undergraduate community is really remarkable, because often out of the mouths of babes, as they say, you often have your most profound insights. And I really enjoy teaching. I think it's my colleagues who don't do a lot of it, I think they're really missing an opportunity, because it forces you to read in areas that you normally don't read in order to stay current. And as I tell my dental students, I will tell them on the first day of lecture, it's about renting versus owning the information. And since microbiology is such a large part of the pathology associated with dentistry, you really have to take ownership of the concepts and the microbes in order to really appreciate how these little creatures that are present at 100 million per mil of saliva actually interact with us and do all sorts of remarkable things. And then if you happen to be a smoker, it actually influences the microbial community, and you get a whole different suite of pathology. So it's really a pretty remarkable event when you sit back and look at it from the perspective of a microbiologist trying to teach the next generation of dentists. And we've had great strides in oral health, and we now appreciate that oral health is so important to our overall health that dentistry is no longer a reconstruction event. It's really now the gateway to healthcare.
Helps us live longer to have good teeth so we can chew, right?
Well, that too. That too. It makes life more pleasurable.
I always think of the ancient humans and pre-humans when their teeth rotted away early in their lives. Probably they couldn't eat.
Yeah, and their life was just misery.
And they died in their 20s and 30s, if that.
Yeah.
So we have a lot to thank. Do you tell your students that every time they chew, they have a bacteremia?
No, every time they brush, they have a bacteremia.
It's
every time they brush. And so if you brush twice a day, you're bacteremic at least twice a day, and it takes a while for your liver and spleen to pull those microbes out. And you really begin to appreciate that dynamic equilibrium between the oral cavity and what's going on with our liver and our spleen and the whole arm of the immune system. And you really then begin to appreciate the importance of things like complement. Oftentimes our medical students and dental students, complement is the thing they love to hate, because it seems so complex, but it's so important, especially for viruses.
That's important for clearing that bacteremia that you get when you brush your teeth, for example, right?
Oh, yeah.
And then do we have people who become very ill as a consequence?
Well, again, it depends on how ill you are predisposed you are. It's like everything else that you are the product of your microbiome and your life experiences as how does your immune system react. And some of the stories we're going to review for our listeners this afternoon really drive home the experiential nature of microbiology and the human condition.
So, Michael, when you have these brief bacteremias, do you make an antibody response against the bacteria?
Yes and no. It depends if it's a new antigen or an old antigen.
So we all probably have a repertoire of antibodies against these oral bacteria that invade us periodically.
And it really depends on self. And really, they're on the outside of us. And, they come in and they get quickly cleared. And our clearance systems really clear them pretty quickly.
So they don't have a chance to multiply at all, right? So they don't probably induce a vigorous response.
Unless you're one of those poor souls who's got a cardiac issue. And then, of course, you'll develop vegetative endocarditis.
Right.
And, that oftentimes is pretty devastating.
All right. Well, I didn't mean to diverge, but this is such a fascinating thing. And I think the microbiome and health and all that is going to be with us forever. So I love to hear about it. Today, we plan to review 10 of the stories from 2011 that we talked about. We do have another guest coming on, so we'll start. And then we'll get his call. The first story that I put on, this is something that I found very interesting, and this was on TWiM number 9 and a follow-up on number 20, and that's the outbreak in Germany this past year caused by E. coli 0104H4. This was, of course, an outbreak of hemolytic uremic syndrome produced by this novel strain beginning in May. At the initial time, the outbreak source wasn't known, but then, as we talked about on number 20, most likely is the sprouts.
And fortunately for me, I wasn't proved wrong. I think one of my first comments was, it's always the sprouts.
That's right. And you said that. I remember.
And I think Elio challenged me, as he always does. And fortunately, it was borne out. And I think what this story taught us is that man has gotten very clever in collecting all sorts of interesting things to eat. And we've been able to process them and deal with them in unique ways. And I think one of the lessons that we learn from the bean sprouts in this novel E. coli is how virulence factors are moving horizontally amongst the species, and how microorganisms are exchanging this information in a very, very almost text messaging, social networking type of manner, in that everybody knows everything about these virulence factors amongst the microbial community so quickly. And we really saw it take root in Germany, and you got to give credit to the German health officials who really, through good old-fashioned shoe leather epidemiology, worked out and traced back the source of the outbreak back to a shipment of sprouts. They actually were able to look at the seeds, and the seeds were implicated, and they were able to contain the outbreak that could potentially have gone global. And I think, Vincent, you asked the very profound question: is will this strain return? So what are your thoughts? You think it's going to be back?
I would say no, because they've identified a source. They'll be more vigilant about that. And if it is detected early on, they'll probably be able to stop the spread more effectively. So I would say no. I think it's sort of like a SARS type thing. We saw it once. It came from bats into people, and it's gone. So that would be my prediction. What do you think?
I think you're absolutely correct. I think the take-home message with this story is that foodborne illnesses have been with humans since the dawn of time,
Yep.
and will be with us well into the future, and we really need to be vigilant. And I think what many governments are coming to the realization is that perhaps they need to develop a more aggressive screening programs for the foods that are traditionally ingested by at-risk groups for microbial infections,
Right.
such as infants, the elderly, diabetics, and we have to be very thankful for health departments like the German health department, our FDA, our CDC, and most importantly, the self-policing activities that the food industry is really taking a leadership position in. This week, we learned of a recall in powdered infant formula for a bacterium Chronobacter, which was formerly Anorobacter, which traditionally caused infections in adults. But this one particular sequence type 4, is a great concern for infants because it results in the typical sequelae are bacteremia, meningitis, or necrotizing enterocolitis. And unfortunately, an infant boy died in Missouri over the Christmas week after ingesting this brand-named powdered formula, which then resulted in a major supermarket chain pulling the brand name powdered infant formula from its shelves. And so they really took it upon themselves at saying, well, we don't know how it got in the formula. We don't know if it's associated with all this formula, but one death was too many. And, it's been causing, looking, through the press, it's been causing infections. And again, I think this sort of situation, like we saw with bean sprouts and now we're seeing with infant formula, is really driving home the need to begin to think about a screening program in which we go after foods traditionally ingested by the at-risk groups. And hopefully our colleagues in the public health sector, in the food industry, will pick up on this and take this problem to task.
Do we know what the source of the Chronobacter was in this formula recall?
No, we don't. I think it's too soon to tell.
Mm-hmm.
We know it's Chronobacter. We know it was associated with the formula. My suspicion is that since it was powdered formula, it's probably in the spray drying process where they make the powder, and there's probably an Anorobacter or a Chronobacter cluster, either bad water or there's some focus in the processing system that they just haven't discovered yet. It's sort of like peanut butter and salmonella from last year.
Yep.
They traced it back to a bad spot in the plant where the salmonella was getting into the peanut butter process. And my suspicion is this is a spray drying process that we well understand, and I think they just have to literally take everything apart and do some good old steam cleaning to be vigilant about getting rid of the bacteria.
Do you think that gamma irradiation of foods is something that would happen in the future, or is that not likely?
People have this phobia of radiation, and we know that it will greatly increase the shelf life because you can effectively sterilize the food so that you remove all microbes from it. But it really brings up the question of, many of us in the microbial community know that our food is not sterile and it comes with normal flora. So the question is the normal flora that we ingest from our foodstuffs important for our good health?
Mm-hmm. Yeah.
And the microbiome was the other big story of 2011, and we really only are beginning to get an inkling of what's going on. And our number 2 story of the year was Helicobacter pylori and its ability to prevent the development of idiopathic asthma.
Well, let's move on to number 2. That's a paper that we did on TWiM number 11. And this was a paper published in the Journal of Clinical Investigation called H. pylori infection prevents allergic asthma in mouse models through the induction of regulatory T cells. Now, we talked on this episode about how the colonization of humans with H. pylori is decreasing over time with increased antibiotic use and greater hygiene, among other things. And in this paper, they show in a mouse model that if you infect the mice orally with H. pylori, this is a human strain, it prevents atopic asthma, that is, allergen-triggered asthma. And the mechanism seems to be that the presence of the bacteria allows for the production of T regulatory cells, which are necessary to suppress asthmatic responses. So this is an amazing finding. Doesn't mean, of course, that it happens in people, but I suppose appropriate studies could be done to address that question, right?
Yeah, and I think, really the most remarkable finding in the paper was the fact that the Helicobacter had to have the virulence factor,
right,
an active form of the virulence factor that is implicated in the development of ulcers that then subsequently develop into gastric cancers. And you had to have the microbe that had the gene that conferred the virulence for the development and then ultimately subsequent sequelae of developing gastric cancer. And so it really sets the whole cancer world on their head, because they were, arguing that we should be thinking about developing a vaccine using that virulence determinant as a target. And all of a sudden, if you would develop a vaccine against gastric cancer based on that virulence factor, then it almost asks the question, will you then predispose you to the development of asthma?
Right.
And so I think it was really a landmark paper, not for the questions it answered, but for the questions it raises.
So you don't think at this point making a vaccine against H. pylori is a good idea?
I don't think until we know more about the cause and effect of, first, why this microbe is being depleted from hygienic or technologically advanced societies. I think that's a very important issue that we have to address before we go off willy-nilly going through the expense and the regulatory, because it's going to probably take $1 billion to develop that vaccine. And would that $1 billion that would be spent to develop a vaccine be better spent trying to understand first why this microbe has disappeared from hygienic/technologically advanced societies, and then secondly, how microbes are vertically transmitted in humans.
Right. This really brings up the issue related to the microbiome that, we feel that much of the microbiome is beneficial, but under certain conditions, they can become pathogens. So here, H. pylori is good for you early in life, but later on, as you age, it may cause ulcers or gastric cancer. And I think understanding why these shifts occur is going to be really important.
And we saw at the general meeting of the American Society for Microbiology a couple of papers this past year in May of 2011 that Helicobacter was also implicated in the onset of Parkinson's. And so this is a really remarkable sentinel microbe, I think, in terms of really defining the human condition, because if you look at asthma, ulcers, gastric cancer, and then Parkinson's, it really has the entire immune system interacting with the microbe as well as our cells in there. And I think this really teasing apart Helicobacter in this dynamic equilibrium it sets up in that extreme environment in the human body. If you think about it, the niche of Helicobacter, which is the stomach, which is it's probably the most extreme environment in the human, with extremely low pHs down in the 1s, the 2s, and the 0s, constantly secreting hydrochloric acid. And then if you consider all the ads we just saw this past weekend about eating too much and the need for heartburn relief, you wonder what these proton pump inhibitors are doing to changing the nature of that extreme environment and what's actually going on.
I'm sure they're altering something. Although the microbiome of the stomach, if I remember from our discussion on TWiM 11, isn't extensive. Is that correct?
That's correct. Helicobacter is thought to represent 50% of the members of the stomach. And so, that's a pretty big number. So I think we're going to work, and if I had to make a prediction for 2012, I think we're going to see more papers on Helicobacter, and our friends who work in the immunology field, especially in the area of asthma, are really going to try to begin to chase this issue.
Well, it looks like our other guest is online, so let's see if I can bring him in. Hello, Dr. Mintz.
Welcome back, Cliff.
Hey, Michael, how are you?
I'm doing well. Happy New Year to you.
Happy New Year to you.
Our third story, this was put in by Michael. We didn't actually discuss this on TWiM this year, but it's a good thing to include. It's the whole brouhaha about H5N1 influenza and biosecurity. And I'll kind of give the executive summary of this. I've thought about this a lot, and I've written about it, and we have talked about it over on TWiV. And I think that our TWiM listeners, some of you who may not listen to TWiV, should be aware of this. It's really important. So, a number of weeks ago, a lab in the Netherlands, influenza lab of Ron Fouchier, announced that it was actually an article in Science about this, announced that it was submitting a paper to Science describing them having taken avian influenza strain H5N1, which is highly lethal in birds and is also lethal in people. They had passed it in ferrets, which is a common animal model for studying flu, and they had derived a strain that was able to be transmitted from ferret to ferret in the lab. And they were submitting the paper describing the process and also the amino acid changes that occurred, allowing transmission. This paper was intercepted by the NSABB, which is the National Science Advisory Board for Biosecurity. It's a committee established in the U.S. after the anthrax attacks to decide whether certain scientific research should be restricted in distribution because of its potential application in bioterrorism. They have since decided that the papers should be censored or redacted when they are published. There's actually another paper by another group, Yoshikawa Oka, which has been submitted to Nature. And the NSABB has said you can't publish the full methods and you can't publish the amino acid changes. So I have argued that their argument is that this will facilitate bioterrorism. I have argued that on many levels, this in no way does such a thing. And I just want to review it, and I want to get your thoughts, Michael and Cliff, on this. First of all, these are ferrets. They are not people. It's well known that what you find in an animal model does not necessarily apply to people. In fact, passing a virus in an animal is often used to make a vaccine, an attenuated vaccine. And it's been done for polio, it's been done for yellow fever, and many other viral vaccines. The lethality of H5N1 seems to scare people, and you often hear this number, it's 50% lethal. There have been a few hundred cases of human H5N1 influenza, but it never transmits among people. But the 50% fatality is kind of misleading because that means half of the people who are admitted to hospitals with this infection die. But we don't know the real denominator for that. And it turns out that if you look in the literature, you can find a number of sero studies which have been done in Asian countries, which indicate that there is a very high level of inapparent H5N1 infection. So it may even be millions. And so the lethality of the virus may in fact be very, very low in people. So this virus has been infecting people for 30 or 40 years, and in all that time it has not been able to be transmitted. So I think that's just another piece of evidence which says we shouldn't really be worried about this. Now, if a terrorist wanted to use this as a weapon, it would actually be an experiment, because we don't know if this virus would be lethal in people. So I don't think a bioterrorist would actually pick this virus. They would pick smallpox or anthrax, something that's known to kill people as well. So there are a lot of scientific reasons why this doesn't make any sense. But maybe more importantly is the censorship issue. They want these papers redacted. And I don't think you should redact science. I think everything has to be published. I think the way science moves forward is that you publish results and people take them and move forward and learn new things. And I think this is a very poor case to set a redacting standard on, because scientifically it doesn't make sense. And I think if you say we're going to censor this, then going forward it makes it very easy for the U.S. government to censor other studies. And I don't think that's a good idea. So that's the background. That's my feeling. Michael, what do you think?
Well, this is an interesting case because this is influenza. And influenza is the original virus that did molecular biology before we even knew what a molecular biologist was. It's the whole issue of antigenic drift and antigenic shift, and it really drives home the issue that we have with influenza and why it's so important to get vaccinated, and why it's so important to continue to fund serum studies in populations in which avian flu is moving through, so we have an understanding of the denominator. And I think this may drive home the issue of how the popular press can actually influence decisions about censorship because of the death coefficient. And we had a similar story on TWiM that we're going to discuss in a little bit about good old E. coli K12 becoming virulent. And E. coli K12 was the lab-tamed strain that was tamed at Stanford back in the last century, literally in the last century, in the early part of the last century. And that paper was published, and there was no outcry at that particular point in time because they described how you could make a lab variant that almost every molecular biology lab has a derivative of K12. And it really illustrates, I think, the problem that the government is really faced with. And we've seen some of these discussions develop over the last few years, dual use being one of them, and censorship of information coming out. And most of what a terrorist who's ordinarily skilled in the art, to use a phrase from the patent literature, will know what to do in order to, if they really want to go after converting H5N1 into a hypervirulent strain. Most of the techniques are already in the extant literature, and I'm sort of with you, Vincent, and I think it's a very slippery slope, because often the best way to develop vaccines is to really understand what mutations are doing. And as you point out, we should learn from our lessons from the past of how we made a vaccine for polio and how we made a vaccine for yellow fever, and why, we go back to the most successful vaccine ever developed by man, and that was, of course, Louis Pasteur's vaccine for rabies. And this was well before the days of IRBs, and and Louis Pasteur developed a very effective vaccine against rabies, and he basically passed it through, and I think it's one of these situations where I think the popular press is influencing the policymakers as well as the scientific community, who say, okay, this will assuage the fears, but in reality, if they really want to do it, most of the how-tos are already in the extant literature.
Yeah. Cliff, do you have a view about this?
Well, I share my view is very similar to both yours and Michael's view, Vincent. But there's one, and everything that Michael said is absolutely spot on, and I agree with you that censoring any kind of scientific information is not prudent, and it doesn't help to advance our understanding of issues that need to be solved, only because science is about sharing data, sharing information, and it should not be down political or nationalistic lines. However, if you look back in history, obviously the Manhattan Project was very much kept under wraps, and there have been precedents set where there were new technologies that had potentially devastating effects like atomic bombs, etc. The space race is another example of where science is held tightly to the vest. But again, we're talking about things that are influenced more by potential devastating events or militaristic or nationalistic needs of the players in the space. But when it comes down to science, particularly in the area of pathogenesis, it's like Michael said and you said, Vincent. Anybody trained in the art really has a good understanding of what can be done, what ought to be done, what shouldn't be done, etc. And my feeling is that this is really to what Michael was alluding to, and something that you and I, Vincent, have talked about regularly over the last several years, is the fact that science literacy is so low in the United States and elsewhere. And frequently the people that are advising the people that are making these decisions are not necessarily the most highly skilled or trained individuals that are doing the advising, because frankly, even if you have a National Academy member advising somebody on the DARPA committee, the person that they're advising may not really understand the advice that they're being given. And unfortunately and very sadly, there are members of the scientific community, both on left-leaning and right-leaning individuals. And I think that in the current environment where there's a presidential election, coming up in 2012, I think the Obama administration, because of what Michael said about the big brouhaha over the strain that we're talking about, that it would be seemingly, if the United States didn't do something, then the Republican Party may turn around and say, how could you not? You're jeopardizing the security of the United States. And I remember way back when, at the beginning of the outbreak, Vincent, you were one of the only few people on the virology blog that was saying, wait a minute, this is not going to be what people expect it to be. And you were very, very clear and very, very cogent in your arguments about why we need a little bit of restraint before we jump to conclusions and start doing things quickly and not carefully. And then we result in the problem of companies developing vaccines that were really not necessary, and then making money off of vaccines that really weren't effective. And then there were problems with the vaccines that were quickly rushed to production. And then the government, was buying up the stocks, and it became a big socio-economic political event, rather than listening to the thought leaders in the field that said, wait a minute, we don't know. So from my perspective, science should never be censored. And I don't really see that if I was a terrorist. Number one, it's been very clear, outside of using very, staid, well-honed technologies like flying airplanes into buildings, most of these terrorists can't even make bombs correctly. The literature for bomb making has been out there for, 50, 60, 100 years. And quite honestly, outside of certain governments, I don't see that there's an active bioterrorism programs going on. So particularly in what you said, Vincent, talking about, and Michael said, we're talking about flu, which is notorious for antigenic drift. So you may figure out something about this particular variant, which doesn't mean anything downstream in terms of its infection rates in humans, as you pointed out, Vincent.
Yeah.
And I don't, quite frankly, I don't understand it, except that the Obama administration was influenced, I think, by people that believe that this was a potential terrorist threat. I think it's completely wrong-headed.
I think this is going to be quite a point of discussion in 2012, so we'll take it up again for sure. I think the basis for this decision is flawed, and I do think science needs to be open. We'll see.
And it should not be influenced by politics. But unfortunately, as we've seen, with the previous administration, where there was an attempt and a pretty good one at denying the notion of, and the scientifically proven or heavily, supported notion that there is climate change, there is global warming, and the levels of CO2 in the atmosphere are higher than they were in the Pliocene era, I believe, and we could be headed for some disastrous events in the future. But the administration, previous one, did not want or did not believe or felt that it would hurt business to start looking at that seriously. And I think that this is another example of that.
There's been a movement now starting to put together an Asilomar-type conference of microbiologists to discuss this issue, the whole issue of what kind of research should we think about restricting or its publication and so forth. I think that's a good idea. I think it has to move outside of this biosafety committee, which I think is already bought into the notion that biosafety is an issue and we need to censor, and I don't think that's the right approach to this problem. Let's move on to number 4. We did 3 different TWiMs on the microbiome. We did one on the effect of diet. That was TWiM 19. We did TWiM 15, the effect of the gut microbiome on susceptibility of the lung to influenza. That was one I really liked. If you take away the gut microbiome or modify it with antibiotics, the mice are far more susceptible to infection with flu, because their dendritic cells don't properly instruct the T cells in the lymph nodes that there's a problem. And TWiM number 2, way at the beginning, the relationship between the gut microbiome, metabolism, and body weight.
And I'd like to point out our colleague who is a regular on TWiM, Elio Schechter, has it on his blog for the last post just before he said goodbye for the year. What's the score on the microbiome? One of the comments that Elio challenged his readers, just for kicks, let's count up recent papers that say the microbiome is a good thing and those that say otherwise. Just count the titles. And one of the comments on his blog was a story that NPR published, and you can go to Small Things Considered, and it's an story that was published in PLOS One about a study outlining mosquito preferences for, this is the mosquito that causes malaria or transmits malaria, for less diverse skin microbiota. And so again, this drives home the fact that the composition of the human skin microbiota affects the attractiveness to malaria mosquitoes. So I think the microbiome is actually making it in prime time. And I think what we really need to remember is our old-time physiology, because it's the physiology of the microbe as well as the host. And I think this PLOS One article on the microbiota attracting malaria mosquitoes, because those mixed microbial end products or waste are actually responsible for recruiting microbes, recruiting the mosquitoes that then will transmit malaria, I think is just absolutely fascinating and drives home how we really need to pay attention to the physiology and the physiology of the host in order to begin to dissect the papers that are going to be coming out in this next year of 2012.
So why do we have this good versus bad? I thought the microbiome was good for you.
Well, the question is, it goes back to the story on asthma.
Yeah.
Asthma, you have Helicobacter pylori, which is good when you're little, bad when you're middle-aged.
Yeah, so it's a double-edged sword then. It depends on the conditions. It's like your oral microbiome. When you brush your teeth, it can kill you, right?
That's right. As they say, everything in moderation. I really think that what we're going to see in probably the next 10 to 15 years is the reblending of microbiologists and immunologists again, like we did in the literally in the teens and 20s of the last century, where microbiology and immunologists were one and the same. I think the immunologists are going to have to relearn a lot of the basic microbiology that they don't think is important. And the microbiologists, similarly, are going to have to learn a lot of the foundational immunology, especially paying attention to both the innate and adaptive parts of the immune system.
As readers may know, or if they don't, you should be reminded that the U.S. government is funding the Human Microbiome Project, and there's a website we will link to. And here, the purpose is to characterize the microbiome of distinct sites: the nose, the mouth, the skin, the GI tract, and the urogenital tract, and determine the composition, that is, the number of the different kinds of bacterial species that are present in normal people, how it varies among person to person, and how it varies in disease states. So this is a very big project, still ongoing, and there's a lot of resources there. All the sequences are published and available for people to look at. So it's a very interesting endeavor, and we're going to hear a lot about this in the coming years because we really don't understand these relationships yet between microbiomes in different people and disease and normal health and so forth. Should be really interesting.
I have a question.
Go ahead.
So when Michael was talking about, the numbers of papers, positive and negative, what are some of the people that are making negative comments about the biome, saying, I can't identify any negative outcomes of doing the work?
So there's a paper there are two papers which showed that two different virus infections are aided by gut microbiota. Mm-hmm. Okay, so that's viewed as a negative because one of them is polio. So apparently polio is stabilized in the gut by LPS, of all things. And another virus, a retrovirus infecting mice, is also stabilized, is bound by LPS, which triggers a immunosuppressive immune response to allow the virus to persist. So those are viewed as a negative, although for the virus, of course, it's not a negative.
Right.
But for the host, it is.
It's supposed to host. So I guess the goal is to reduce the number of gram-negative bacteria that are in your gut microbiome. Is that the one would surmise from those papers? Notwithstanding the fact that most people, at least in the United States and globally now, are vaccinated against polio.
Well, it depends how generally applicable these findings are. I mean, if it applies to, say, rotaviruses, which are much more frequent now, or other gastrointestinal viruses, then it may suggest an intervention. But we have to see that first. I don't know.
So it's really not a negative that it shouldn't be done. It's more of like there's negative results.
Yeah, there's a negative consequence of having the bacteria there because they aid in viral infections.
But the bacteria have to be there anyway. So
yeah, that's right. You can't get rid of them.
The more you know, the better informed you are. So I say those are great. Those are very important publications.
So if the whole world were a notobiotic,
right,
what would happen? We'd probably be okay, right?
We'd have a very dull diet, though.
We'd have a dull diet.
We'd all die. We wouldn't have any vitamin K.
Well, but notobiotic animals don't die, right? So they live, but they live, as Michael says, they have poor life.
They have a very bland diet.
I mean, the argument that nobody in nature is notobiotic is kind of weird because there are bacteria that colonize everyone, so you can't be notobiotic. So,
but the thing is, Vincent, with notobiotic animals, they're fed sterilized food, kept in right.
Right.
So they're a specialized set of creatures that can be kept alive.
Yeah.
But I think it really drives home the importance of using gnotobiotic animals to do some of these studies, because what Cliff brings up is a real interesting point, because you can add back if you have you almost have a defined experiment with a gnotobiotic animal where you start out and you inoculate the animal with a defined microbiome, and then you can watch and learn what's actually happening, because the animal does indeed have an intact immune system, unlike some of these synthetic knockout animals in which they have bizarro immune systems. I mean, it's truly from bizarro world. It could almost make a comic book out of the immune system of some of these knockout animals that are being used to study many things. And I think gnotobiotic animals, because they're, quote, normal, you got to remember humans are a very outbred species, with the exception of royal families. So
that's right. That's
right.
Right.
So it's a rather interesting issue that you brought up.
I think we need to get some microbiome researchers on TWiM this coming year, so they can tell us exactly what's going on here. I think it would be very interesting. But this is a big topic, clearly, and was in 3 of our podcasts. So there you go. Number 5 was a paper we talked about on TWiM, number 12, and this was an amazing paper in Science where they changed all the stop codons in the E. coli genome by genetic engineering. A tour de force of genetic engineering. You really liked this one, didn't you, Michael?
Well, I sort of drew the short end of short straw to tackle this. This was one of these wonderful science papers that you have to read all the supplements to really appreciate it. And our good friend Elio cajoled me into writing a guest blog post to his routine blog, Small Things Considered. And I did it, and I was actually very honored that he put me in his top 10 of the genomics and evolution piece of, and the title of the piece, What's Old is New, Genome-Wide Manipulation of the Bacterial Chromosome in Vivo. And people who were trained in the area of us, Vincent, Cliff, and myself, we learned how to do this God-fearing type of genetics with these crosses. And they just took this to a whole new level of making these crosses, and it was really a remarkable piece. And Elio and I went back and forth about how much of the old genetics I should put in, because I appreciate that many of my younger colleagues don't appreciate the Beckwith type of genetics that we've learned, and the crosses that we did. And so I encourage the listeners to go and take a look at that blog post, because I distilled it down, as well as really take a careful look at that science paper, because it really is a primer on how, if you want to do something on a large scale, at least in bacteria, how it's really possible. But you have to remember your old microbial genetics and all the caveats and why some of those strains are in the back of those cloning catalogs that you can buy your restriction enzymes from. Take a look at the strains and all those little caveats that they have next to those strains, and the mutations that they are all carrying are there for a reason. And that's what this group out of Harvard and MIT really exploited, is that those strains, in order to really exploit this. And I think synthetic biology is going to be another area that we're going to see a lot of in the coming year, because energy is, of course, very important. And we know Exxon and Craig Ventnor are actively exploring making the ideal strain to make oil or to make motor fuels from bacteria. And they're probably taking many of the lessons that were described in that science paper and putting them to work.
So it's going to be a generally usable technology that they used in that paper that they illustrated. They were trying to change all 314 TAG stop codons in E. coli. And if I remember, they didn't do all of them, but they got pretty close and they worked out the methodology. It's not easy.
They almost got there.
Yeah.
They almost got there. There were a few crosses away, but after you do so many crosses, you're either going to go to the Jack Daniels or you just give up.
Great. All right, number 6 on our list has to do with probiotics. TWiM 24, which explored the use of probiotics to treat intestinal diseases, probiotics and inflammasomes. We had 2 papers, one describing a soluble protein ameliorating intestinal inflammation, and then the regulation of colonic microbial ecology and the risk for colitis by the inflammasome, which is a immune complex. Probiotics, and Michael wrote in the show notes, more hype than science? Why is that, Michael?
Well, we started talking about what school I'm going to be teaching come next week, and I'm going to be talking with my future colleagues in the dental community, and probiotics have been really being advanced as a therapeutic in the periodontal space for quite some time, and with very little evidence of their utility other than there haven't been major studies. There have been limited studies, but it really hasn't been major yet. I think the 2 papers that we selected this year to talk about probiotics really begin to lay a solid foundation that probiotics are there's really solid science behind the utility of using a probiotic to limit a inflammatory response, which periodontal disease is, as well as the diseases that these were specifically looking at, namely colitis. And it really begins to teach the community of how to begin to evaluate many of the probiotics that are in the marketplace today. Many of us probably have seen in the normal television shows ads for probiotics that are being produced by major food companies and some drug companies in which they're advocating colonic health. And, one of the things we try to teach our medical students and dental students is evidence-based medicine, and is there real evidence behind some of these claims, because our patients are often coming to the physicians and the dentists and asking them the question, will this actually work for me? And so you have to be able to evaluate whether or not these things really work, and I think these papers are now beginning to show, at least from a mechanistic perspective, that there's real science behind this, and we're going to have to do careful case-controlled, placebo-controlled trials in order to really understand how well probiotics will actually influence and improve our health.
I think you have to go. These 2 papers were excellent because they had very specific components. One, they had a protein from a specific bacterium that they were using. And so this is well beyond taking yogurts, for example, or probiotic preparations, and we're not even sure if the components are useful. So that it seems to me that is what's going to have to be investigated, what particular species you need to correct whatever syndrome you're looking at.
It goes back to freshman chemistry. It's the limiting reagent problem. What is the limiting reagent that is doing the good thing?
Do you have any opinions about this, Cliff?
Well, I think that there's a general resurgence in looking at both probiotics and natural products for their ability to provide therapeutic applications and various things, mainly because Historically, anecdotally, when you look at traditional Chinese medicine and traditional Asian medicine, or, even in South America and Africa and stuff, there's always been a kernel of benefit or truth to some of the claims. And I think given the state of the food and pharmaceutical industries, I think, put simply, they're running out of stuff to actually market and sell. And I think that they are beginning to look at things that previously were probably not worth it from them in terms of return on investment, to begin to take a closer look and actually use evidence-based medicine to either verify or refute the benefits that some of these probiotics and natural products have to offer. And I think talking with some of my MD friends that are in the clinic, basically when you're dealing with patients these days, you really do have to ask your patients, what else are you taking besides the prescription medicine that your doctor has suggested that you take? Because so many people these days are taking so-called natural products, probiotics, because they're either sick or they're at risk for being sick, and they believe that and perhaps, I used to be somebody who was a naysayer, but more and more when you start looking at the literature, and I have been recently looking at the effects of the breakdown product of ginsenoids, which is compound K, which is a metabolite that is actually, the ginsenoids are transformed by gut flora into this identifiable molecular species called compound K, and they're looking at compound K and purified, or as pure as they could get it, as a treatment for oncology indications, or possibly for treatment for even statin or cholesterol modulation. So there's all these things that people like. Ginseng is a root that people in China have been eating for thousands of years, and according to my Asian friends, at least Chinese friends, ginseng is the king of all herbs, that if you take ginseng and eat it regularly, just the root, your health is going to be much better if you don't. So people have been doing it for 5,000 years, and now they're actually looking at the science, possible science behind ginsenoids and why they work, and they're doing it in animal models as well as controlled clinical trials like Michael suggested. So.
Cool. Science moves forward. That's great. I'm sure we'll hear more about that. Our number 7 is something I really enjoyed the past year, Symbioses. We did 4 different TWiM episodes on Symbioses. TWiM 21, the latest one, on Patacatenula, this symbiosis in worms, these worms that are just full of bacteria that help them to survive. And then TWiM number 17, 2 different symbioses, one between rickettsia and whiteflies, and another one in mealybugs. And that one is so cool because it involves nested symbionts. That is a bacteria within a bacteria within the mealybug. And they all have incomplete metabolic pathways that they use to complement one another. Number 10, the cloaking device utilized by the tiny squids that are full of Vibrio that make light and allow them to cloak themselves in the waters when the moon is up. And Elizabeth McFall-Nye was, of course, that's her work in her laboratory, and she described that beautifully. And then back in TWiM 8, when we did a live episode at the GM in New Orleans, we heard about the tube worms down by the deep vents on the ocean floors, and also in shallower waters as well, which are also full of bacteria that provide them with what they need. That's the work of Nicole Dubilier. So I'm really fascinated by these symbioses. Maybe it's because I didn't really read much about them previously, but I suppose they're all around us, and the more we look, the more we'll find. So am I too enthusiastic about these symbioses, or are they really neat?
I think they're really fascinating, and I'm especially impressed with how a rickettsial infection can alter something that I took as foundational to species that have sex, the ability altering the male-female ratio,
Right.
and how a bacterium can actually be used to exploit an insect to actually have an effect in agriculture. I mean, this is really a pretty remarkable story of how microbes are interacting with the eukaryotic, hard-coded, if you will, program, and altering the way it behaves. It was just absolutely fascinating, and I think, this family of stories in concert with the microbiome, I think in a few years are going to merge together, where we're going to have a sophisticated set of tools that will enable us to answer complex questions, because the symbiosis, it's really a series of complex questions, and Margaret McFall-Nye's Cloaking Device story I found was absolutely fascinating as well. And that same virulence factor family that we talked about with Helicobacter comes back to visit in the Vibrio that she's looking at. It's the same sort of family of virulence factors, but it's having a very interesting biological effect on how the squid dumps the, if you will, the microbiome and then collects it back.
Yeah, it's just great.
It was just
Beautiful.
Wonderful. It was like a trip to the zoo without having to go to the zoo.
I think I called her Elizabeth. I'm sorry, Margaret. I do that. Margaret McFall-Nye, who is on sabbatical, and hopefully when she's finished, will join us back on TWiM again.
I'd just like to say one thing.
Sure.
I've always had a healthy respect for symbiosis, and I think
it's good.
just because everything comes together and it's all part of an ecosystem. But symbiosis relationships have always been, really, really important for the organisms that are in those relationships. But I think the reason why I've always had a healthy respect for them is that, again, back to my graduate training at the University of Wisconsin-Madison, there was a guy named Winston Brill there who was heavily into the rhizobium legume interaction. And once you learn the physiology of the interaction between rhizobium and alfalfa root or legume root nodules, you begin to understand how bacteria could really change the physiology and functioning of leguminous vegetables. And that's when I realized that bacteria frequently get a very bad name because everybody knows that they cause disease, but they do a lot of very, very, very beneficial things, primarily through symbiotic relationships. So I've always been really, really fascinated about how the adaptive pressures are brought to bear so that the both species benefit, or at least one may get a little bit more than the other, but everyone's happy at the end of the day. So it's a great, I think it holds a lot of promise, and again it goes back to why we should fund basic research.
If bacteria would just stop killing people, they would lose that bad rap.
Yeah.
Well, if we develop enough vaccines like they do in virology, then that may someday come true, Vincent.
Well, I'm sure we'll see more symbioses in the next year as well. Number 8, a story we mentioned previously, turning E. coli into an invasive organism. This was a paper in MBio from Shankar Adya in Carr's laboratory. And this was amazing. 2 amino acid changes in E. coli change the supercoiling of the genome and completely change the transcriptional program such that cryptic genes are now expressed that allow the bacterium to invade epithelial cells. Who would have thought that could happen?
Well, certainly not the folks applying to my biosafety committee, because everyone who ever writes an application describes to me how K12 is a very benign microorganism, and these two mutations, because it's altering the superhelicity of the genome, it really drives home the importance of understanding how genes are placed in context. And I don't think you really learn this lesson until you do enough cloning and you really appreciate how important superhelicity is to whether or not your gene is really going to be expressed at the levels it is. And you only have to talk to people who worked in some of the early genetic engineering based companies to where they were trying to overexpress truckloads of a particular protein, and you listen to their horror stories and how they had to tinker with the physiology in order to get things to go. And it could have just been of where they put their construct in the plasmid that they were using to create things of how the promoter was effaced or what actually was going on. And I think this story really drove home how naive our understanding is of what makes something virulent. And I think that really is pretty neat.
I liked the fact that E. coli, which, as you said, everyone thinks is benign, clearly has a set of genes that at some point, somewhere in some host, may be used to allow it to be invasive under some conditions or to do something that we're not aware of. So that's the cool take-home that I had from that.
Yeah.
Maybe they should have redacted or censored that paper.
That's right.
Because that would have more.
That's right.
That would have more far-reaching effects than the flu.
When we were talking about the flu, I wrote down in the show notes is that this particular story ties in very tightly with story number 3.
Sure.
But when most people think about K12, they really don't think of it as this three-eyed monster that it could potentially turn into.
Right.
Well, when you think about it from an effectiveness standpoint, in terms of, not I don't want to get, the police, coming to my house, but if you could introduce those mutations into all these garden variety E. coli and then, sprinkle them into food or whatever, drinking water, they'll colonize, most people's intestinal tracts if they've got the fitness, and you can wipe out really large numbers of people from, dying of enteroinvasive E. coli infections. Sure. I mean, it's just, to me, that's got more of a likelihood than transmitting a mutated flu virus to one susceptible person to another. So yeah. It's all relative, right, Vincent?
Sure. No, I think the point is that there's so many ways that you can make pathogens that it makes no sense to censor any of them.
Right.
The 9th story is one that comes from the latest ICAC Live, which was in Chicago. And there, Arturo Casadeval told a story about his theory of why we don't have more fungal infections. And I found this really interesting. So there are thousands and thousands of fungal species on the planet, but humans are infected by very few of them. And the theory is this is because our body temperatures, 37 degrees centigrade, don't permit the growth of most fungal species, which prefer lower temperatures. And so he is trying to address that hypothesis. And one of the examples he gave in support of it is that there's now a global extinction of amphibian species by a chytrid fungus. And this makes sense because these are amphibians with lower body temperatures than ours, and the fungi that are infecting them have no problem replicating at that temperature. So I found this really interesting, and I look forward to maybe seeing it tested in the near future.
I think it really drives a critical question to our understanding of our sense of place and asks the question what climate change is doing to our exothermic, cold-blooded reptilian friends. And so, again, I'll throw down the gauntlet to those people out there listening who may be interested in asking this question is, have we begun to monitor changes in the fungal biome in cold-blooded reptilian friends, where we've actually seen in some places like Greenland and Iceland, we've seen 4-degree deltas in temperature over the last 30 years, and so you could go off and monitor reptiles, and even in such places as the Amazon, I think they've actually seen temperature changes, and you begin to wonder whether or not the fungal biome is changing in this population, and whether or not it's selecting out a more virulent, or again, these microbes have evolved in concert with the reptiles, and as the climate change is driving temperature changes, is it beginning to cause problems in the fitness of these populations, as we're seeing climate change on a very real scale?
Good question. Maybe Arturo would care to speculate.
Yeah. Next time I see him, I'll ask him.
All right, that brings us to number 10. And I thought that I loved our friend Al Sacchetti on Microbiology 911, number 22. So Al is an emergency room physician over in Camden, New Jersey, and he talked about real-life microbiology, what kind of infections he sees, and I just think that's great to bring it down to reality now and then. So that was one of my favorite episodes.
And as it was mine, I always enjoy talking with my clinical colleagues who sort of bring us card-carrying microbiologists back to reality.
That's right.
And Al certainly did that. I mean, he taught me something that I hadn't learned, is that Caucasians actually are more predisposed to UTIs than people of color. And that for me was truly eye-opening. And he was just an absolutely fascinating guest.
He's
and
a good guy. Yeah, he's a good speaker. He has a lot of experience, and we'll have him back more. And in fact, we have an email moving into our email phase from a listener referring to Al's episode. So let's read that. That's from Jane, who writes, "Thanks so much for your podcasts. I find I am a person who learns best audibly and have just jumped into the science, history, medical, and EMS blogs. I listened to a podcast called Smart EM by a pair of emergency room docs, and they covered the issue of urinary tract infections in peds. They do journal and study reviews, and they found that there isn't any basis for the idea that untreated UTIs cause kidney damage for peds. I know, but they took the time to ferret out all the info. Anyway, I'm enjoying your podcasts. I don't think going back to school is something I can do, but about once a week, I go to a class on virology, parasitology, and microbiology. The tuition is great. No student loans. No sleeping in class. Thanks."
What a wonderful comment.
Good points, right?
Yeah.
So I sent this over to Al Sacchetti, who answered right away. Jane is correct and not correct, depending on who you read. This is one of those topics that will continue to be debated, as it is mostly soft science. It is all based on retrospective case studies, so you really can't design what would be considered a good scientific study to prove it. Such a study would involve not treating a cohort of children with urinary tract infections and following them and a second subset of treated children to determine if one group develops more hypertension than the other. Not an ethical study to perform. The other confounder is that there are multiple other causes of hypertension that may overwhelm the cases caused by infant UTIs. For me, the take-home message will not become clear for another 2 decades. That is how long it will take for this generation of treated children to grow old enough to really look at the epidemiology of their hypertension. As an aside, it is one of those issues that really will not change our practices, since we will always treat a UTI in a child. On a different note, what would you think about doing a TWiV on Hep C with a Hep C virologist, a transplant surgeon, a hepatologist, and possibly a Hep C patient, from the bench to bedside type of show? Take care and continue the great work you do with all the TWs.
Well, I think Al really, drove home the importance of good epidemiology, and it's something that I think will come as we get a comprehensive digital medical record. The PEDS generation, the ones that are considered pediatric patients today, that are in this cohort developing these UTIs, will probably be the first generation to have a comprehensive digital medical record. And then in the years to come, we'll be able to decipher whether or not those children did indeed have an untreated UTI, or what it was treated with, or something along those lines, and we'll see whether or not it works. And it will go from a soft science to a hard science. And, the way you go from soft science to hard science is with more data.
Yep. So thanks for the reply, Al. And I think your idea of a bench-to-bedside TWiM would be great. It's just a matter of getting all those people together on one show. It's a bit of work. Our next email is from Al. Hi, guys. I was wondering if you had seen the following article about the lack of acorns being produced in the Northeast this year. In it, they mentioned that the lack of acorns in the past is associated with crashes in the mouse population. And then the author says, and because the now overgrown field mouse population will crash, legions of ticks, some infected with Lyme disease, will be aggressively pursuing new hosts like humans. So he links to a New York Times article about this called After Lean Acorn Crop in the Northeast, Even People May Feel the Effects. Then he continues, currently here in Atlanta, our oak trees are producing massive quantities of acorns. Every few years, oaks will begin producing lots and lots of acorns in a season, a phenomenon thought to saturate their predators and try to increase the odds that some of their seedlings will germinate and take root. Is there any way that you could do a show or part of on some aspect of this oak mouse tick Borrelia story? It has the parasites, ecto, tick, endo, Borrelia angle, and it has this beautiful tie-in to local ecology. I know it isn't typically what you do on the show, but if you can do some aspect of it, that would be great. I'm going to use this story in my non-science major introductory biology course next semester for some active learning in the classroom, and being able to have my students listen to another podcast as preparation for those classroom sessions would be too perfect. Does Dr. Gwads have an expertise on the ecology of Lyme disease, maybe? Thanks for the great shows and keep up the great work. So Jeff is at Georgia Perimeter College.
Well, it's another bench-to-bedside sort of story.
Yeah, it's true.
Except it's going from tree to bedside.
Yep. I think this is a cool story, and we will, Jeff, try and get someone on who can talk about this on one of our podcasts. I'm very interested in it myself, and maybe Dixon knows some people who could do that. Acorns and ticks, very interesting.
Wasn't there, looking at the rodent population, isn't that how they think the Hantan virus in the Four Corners area bloomed?
That's absolutely right, yes. So there was a good pinon nut crop.
Yeah.
So you make a lot of mice, and it gets into people's houses, and the virus is in their urine, and then you inhale it, and you get Hantapulmonary syndrome, right?
Yeah, so I think you already we have evidence that does indeed happen with the Hantans, and so I think it's not that much of a leap to think about it in Lyme.
Not at all. It's a really good idea. I think a show on that would be great. So we'll try and do that. Well, that will do it for the last TWiM of 2011. You can find TWiM at iTunes, at the Zune Marketplace, and at microbeworld.org/TWiM. If you do subscribe in iTunes, please go over and leave a brief review. It helps keep us on the front page of the podcast section, and that way more people will find us and learn more about microbiology, which is what we're trying to do here. We like to get your questions and comments. Please send them to twim@twiv.tv. Cliff Mintz can be found at biojoblog.com. Thanks for joining us today, Cliff.
Thank you, Vincent. It was a pleasure. Good talking with you and Michael again. It was fun.
Good. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Cliff, and thank you, Vincent, and Happy New Year to everyone listening.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, Chris Kondayan, and Ray Ortega for their help. Happy New Year to all of our listeners. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.